Mm-hmm. Okay, then let's go. So it's my pleasure to talk today with Dr. Darryl Irwin, Vice President of Scientific Affairs of Agena Bioscience. And basically, we will be talking today about Agena technology and Agena application. So how it works and how can we use it. I hope that now Darryl will tell us about Agena in like broad sense. But again, in the first place, I would like to somehow sum up my current understanding, then just to later to test it against the golden truth. Okay, so my understanding is that Agena is using MassARRAY technique. So that's basically the main technique it's using. So the technique allows you to profile SNP variations in the DNA using mass spectrometry. So that technique is around for quite some time, so a couple of decades, I think. So probably it was a bit in the shadow of NGS lately, but basically now it's coming back in a niche of cost-effective low to medium throughput screens. So that's how I see it. Am I correct?
Yes, largely you are correct, Eugene, and it's nice to talk to you today. So the Agena technology is called the MassARRAY system, and that is a MALDI-TOF mass spectrometry based system for analyzing nucleic acids. And we look at a variety of different types of nucleic acids and nucleic acid changes, including SNPs, whether they be inherited SNPs from the germline or whether they be somatic variations in cancer. We also look at fusions that might occur somatically. We look at methylation markers, gene expression profiles, and so on and so forth. So really it's a toolbox for looking at any type of nucleic acid, because what mass spectrometry does is it detects and weighs the mass of a fragment of nucleic acid. And by knowing its mass, you know what its sequence is. So most other technologies use fluorescence. So they use a primer that has a color probe attached to it, and they make an assumption as to what that's being bound to. But with mass spectrometry, we're not using fluorescence. We're detecting the nucleic acid itself, and from its molecular weight, we're determining its mass. So you're right, the technology's been around for around about 20 years. Agena itself was formed 7 years ago, and we've improved the technology, particularly improved the workflow of the technology. So that is, lots more walk-away time, lots more automation, particularly in the sample preparation side, but also in the data analysis side. And what we've also done is we've seen an evolution of the technology. As disease requires more biomarkers to be assessed per patient, there's a need to do this sort of mid-plexing, mid-density analysis, looking at a variety of different biomarkers to determine, what's the source or what sort of treatment can be applied to that disease. So that works very well with mass spectrometry, where we can multiplex anywhere from just a couple of biomarkers all the way up to 40 to 50 biomarkers per reaction. So that's the sort of evolution that we've seen over the last 20 years with the technology.
Okay, and what about samples? Like, are there any restrictions? What material can we use? Like maybe FFPE samples, other clinical materials?
Yeah, we use highly degraded samples all the time, because we keep our amplicons very short so that they're robust if the DNA has been broken up, as is the case with FFPE, as is also the case with cell-free DNA. So we're quite robust to all different types of input nucleic acid, so input DNA. But also we can detect down to very low copy numbers. So we've got papers out there where we can just amplify from a single cell or just 2 copies of DNA. So then it comes down to, all right, well, how much DNA do I need for the application that I'm trying to achieve? If I'm trying to detect a somatic mutation that might be at a 1% frequency, well, I can't put 2 copies in because I can only ever detect 50% minor allele frequency with 2 copies. So it then becomes a question of, in order to achieve the limit of detection and the sensitivity that I need, this is how much DNA mathematically I need to put in to do that.
Is it possible to go like single cell, because it's probably the cutting edge in NGS now, and I might imagine some applications if you can measure, let's say, 50 markers per cell, you can like maybe make a time points in a tumor patient, for example, initially sequencing by NGS and then looking how the tumor basically evolved. So do you have something in mind like this?
Yeah, we're doing some work with a couple of different groups looking at single circulating tumor cells at the moment. We've previously done work looking at just single sperm, which of course single sperm is just 1 copy of DNA and not 2 copies. So that's getting down to the real minimum input of just a single copy. But yes, we've got a methodology where we can take a circulating tumor cell, lyse it open gently, and then amplify it and look at those markers in that cell.
And how customizable are those assays? So as far as I understand, you basically sell some company-made panels, but you also advertise that one can create its own panel. So how it's basically works? Can I create my panel in the laboratory, or do I need to order it from you? Like, what is the time of developing and so on?
Yeah, so we have 3 different options available to our customers. So the first is we have a variety of off-the-shelf panels, which have content that is generally relevant to most of our users. So frequently that content is in industry guidelines or clinical guidelines. But if those off-the-shelf panels don't meet your need, then we have our Assays by Agena group. And Assays by Agena uses our internal skill. We've got about 16 scientists in the team, very experienced in developing custom assays, and they design your specific content and verify it on control material and send you a robust working panel. Now, the third option is that we do also have our assay design suite. So our customers get access to a custom assay design software where you can put in your own sequences and design your own panel and order your own oligonucleotides and run it on the general purpose biochemistry. So all 3 options are available. And although most groups just prefer to outsource it to us because we are experts, and our turnaround times are anywhere from 4 to around about 10 weeks, depending on the complexity of the panel.
I just have to ask, like, are there any difficult cases where you are basically not able to make a probe for some SNP which lies in low complexity regions, or maybe repetitive sequences? So are there like any?
Yes, so there are always challenging regions in the human genome, although our approach, because we do PCR amplification followed by single base extension, so we have 2 layers of specificity. We're using endpoint PCR, but we're still quantitative at endpoint via the methodology that we use. But endpoint PCR allows us to push those reaction kinetics all the way to completion. We actually have a very high conversion rate from custom sequence that you wish to be designed through to robust assay. In fact, it's one of the highest conversions into robust assays of all technologies on the market. There will always be complex regions. And sometimes we can overcome them with our expertise. And in very rare instances, we won't be able to design it. But usually that's identifiable up front rather than known after the process of trying to design it. Our assay design software gives us the appropriate warnings. We can look and use our expertise at how we can optimize based on those complexities and give our customers that advice at the front end.
Sounds quite nice, because okay, of course, when you're working with QWERTY PCR, you probably order a couple of pairs of parameters beforehand. And here you are bind to this SNP you are going to test, and there is not much you can do probably about the sequence.
Well, exactly. And multiplexing is quite a challenge with RT-PCR because each of the assays have different kinetics, but our approach allows us to multiplex very efficiently without substantial amounts of optimization.
So basically all your primers works more or less in the same temperature and...
Correct. Yeah. We generally use the same reaction kinetics and the same thermocycler protocol, so that you can actually have multiple different panels on the one plate, because some customers don't want to fill an entire plate with just one test. They might want to have, part of the plate testing for NIPT, part of the plate testing for something else, and that's capable on the system as well, because it's the same amplification conditions for all panels.
Okay, and basically the company sells only the mass spectrometer itself, or you also sell some other equipment for sample preparation?
Yeah, so the MassARRAY system is the mass spectrometer as well as what we call the chip prep module. And that module takes the thermocycled plate, conditions it, transfers it onto the mass spectrometer, and then the mass spectrometer does the analysis. And we also sell the panels and the biochemistry to run those panels. So what's needed in the middle there is a thermocycler and either liquid handling robotics or pipettes. Yeah, but with our workflow, it's what's called a homogeneous workflow, which means that you put the sample into the plate and then everything stays in that same plate. You're not doing any plate swaps, plate transfers, cleanups, or anything like that. So it's actually, it's possible to do it just with manual pipettes, but some higher throughput facilities use some liquid handling robots, and it's capable of that too.
I don't think that you would like to pipette like 384 well plate manually, so I suppose you have to have a robot.
Well, we have I've got the laboratory just over here, and the technicians and research assistants manually pipette 384 plates all the time. It becomes second nature. But I agree, higher throughput laboratories like to have liquid handling robotics in order to automate that. So
let's then move a bit to the real world applications. Like, what are the products you're currently selling, and what are the main areas your customers using the Agena?
We have designed all variety of different panels. So focus areas for us are in inherited genetics. So what we're talking about there is things like cystic fibrosis, thalassemia, so other markers like that. And also in the inherited genetics area, we do a lot of work in pharmacogenetics. So that is looking at the metabolism enzymes and determining whether a drug is going to be effective in a particular patient or not. Then our third focus area is oncology. So that's both tissue analysis as well as cell-free DNA analysis. So looking at circulating tumor DNA. And then we also have a variety of panels across methylation as well. So there's things like MGMT, MLH1 are clinically relevant in the methylation space.
And for example, what is the size of your pharmacogenetic panel? How many markers are you testing?
Yeah, we've, again, a variety of different panels that we offer and also custom capabilities there. But our Veridose core panel, which is really the workhorse that we utilize for pharmacogenomics, Veridose core covers, I think it's 21 genes and about 74 different markers in those genes, and it's a 3-reaction panel. And then we have an extra well that does the copy number of the CYP2D6 gene, because CYP2D6 is involved in metabolism of all variety of different drugs. And it also has copy number variation, which needs to be utilized in addition to your haplotype status to work out your metabolizer status.
And what are the main customers? Is it like academia or are those clinics? So
yeah, well over 90% of our customer base is clinical laboratories. So they're clinical laboratories that either that develop laboratory developed tests. In the United States, they are CAP clear laboratories and different levels of regulation in other countries, but they're these accredited clinical molecular pathology laboratories.
Okay, so it's not focused on academia research mainly, or
we have a variety of academic customers, and we support those academic customers. The instrument is used for anything from genotyping plants through to livestock to doing other research studies. So it's still applicable there, but where we've seen the greatest application is in the clinical space.
But by the way, I looked through the products on your site and saw the COVID tests.
Which one?
Test for COVID-19.
Oh, yes. So yes, the SARS-CoV-2. So yes, exactly. This year's been, well, last year now, isn't it? Last year was an unusual year. SARS-CoV-2 emerged and caused the COVID-19 pandemic. So we rapidly developed a SARS-CoV-2 test.
So basically,
and again, we utilize our multiplexing ability. See, one of the major concerns with SARS-CoV-2 is that there's mutations. There's strains that are emerging, base changes that are emerging. So our panel has 5 different targets in the panel, so it's very resilient to potential mutations, and that's very topical at the moment, these new strains that are emerging. So we've got the SARS-CoV-2 panel, which is it's FDA emergency use authorized, CE-IVD marked in Europe. And we've also then developed a new panel, which is a research use only panel, where we've added influenza A and B in with the SARS-CoV-2 markers.
And how long did it take for you to develop the test? Like when it was out there in the market?
Yeah, well, it was quick. We started development in March and we finished in June. So it was a sort of less than 3-month process.
But I think since the summer, some new strains basically appear, like UK strains. It's quite recent. So does your panel work with it? Are you planning to extend your panel?
Yeah, so we're robust against all of those variants of concern, and we're not compromised by those mutations that have evolved in those strains. But also we're currently developing a variant of concern panel, and we've got a webinar on that in just over a week's time that people can access through our website. Because that's becoming more of interest, is identifying those strains, not just detecting them, but identifying which strain it is.
Yeah, exactly. That was my question. So could you identify strains?
Yes, we can.
So probably let's move to the NIP, which is supposed to be our main topic. So there are quite a lot of work was done in NIP, and I think there are some pretty strict regulations in this area, and probably might make sense to talk a bit about this. Like for example, how you're going to feed. So as far as I understand, the Agena developed its own NIP test. Also, I have not found any information on it so far, like on your website or something.
Yes, so this is a very new test, and we're approaching this via a partnership model. So that is, we're enabling partners in various different countries, including Russia. We're enabling those partners to do their own validation, validate, register, and then market that locally. And those partners are at various stages in the process. So we're not doing a global launch, but rather we're enabling partners in specific countries and geographies to validate and register the test. So as those get validated and registered, you'll hear some more, but we haven't launched it as such on our website.
Do you provide your partners with some like experience, advices, supervision?
Yeah, exactly. We provide them with all of the reagents, reporting and analysis software, instructions for use or workflow manuals. We've also done a variety of internal validation studies, so we provide them with all of that information as well. So, but what they do is they then take that and they do a clinical study in their own country and then validate, register it with the local authorities in that country.
I see. So, basically, why do you need yet another NIPS test? I mean, there are like there are basically obligatory ultrasounds and biochemical tests which women have to take. So how you would fit your test, and why do we need a new one?
Yeah, well, ultrasound and biochemistry has a known false positive and false negative rate. So it really is sort of a broad screening strategy. And then you move into the NIPT technologies, which generally have better sensitivity and specificity. But the current technologies, they use a big data approach. So that is, they generally sequence huge amounts of material and generate huge amounts of data. And the effect of that is that they have very high cost. So they've been targeted at the high risk populations. So our customers have been asking us, can you come up with a lower cost, higher throughput screening method that we can apply in front of those big data approaches to better filter our population for those that are at risk in the low risk population. So those that are at risk because it looks like they have an aneuploid pregnancy, even though their maternal age or other factors may not have been indicated they're at high risk, in order for only those samples to go through for that expensive sequencing approach.
And so basically it's kind of a pre-screening, let's say.
Yeah, that's right. So we've run well over 2,000 samples now, and our sensitivity is at well over 99%. Our specificity is at 94%. So it means that we out of those patients that we pre-screen, we capture the very vast majority of all aneuploid pregnancies of trisomy 21, 13, and 18 is our focus area. We detect the vast majority of those, and only and our specificity is very high at 94%. So we put this very enriched population through to those expensive NIPT approaches.
And those impressive numbers are those for each of the chromosomes you are testing, or because I know that in other tests probably chromosome 13 is a bit problematic and the specificity lower there. Do you have any?
Yeah, that's for each of the chromosomes. So we're looking at 13, 18, and 21. We have had access to the trisomy 13 and 18 samples in order to generate those numbers, but of course they are rarer pregnancies. They're rarer instances, so lower numbers of instances. But that's across the entire panel.
Actually, I know that I think that you have done your PhD in the area of non-invasive prenatal testing. So
I did. Yes, I did do my PhD in the area of non-invasive prenatal testing, and in fact, I had the opportunity right at the end of my PhD to use the MassARRAY for that approach. I was isolating fetal cells from pap smears, so very different to the current approach of taking it from the maternal blood. But yeah, very experienced in the area.
How's basically field progressed since?
Well, substantially. I mean, when I was doing my PhD, we were looking for fetal cells, both in circulation and in the cervix. And really the identification of circulating cell-free fetal DNA in maternal plasma, it caused the area to evolve very rapidly.
And basically, what methods are you using to extract fetal DNA from the maternal plasma? So how?
Yeah. So that's where we differ to the other tests that are on the market. So the other tests basically take all of the cell-free DNA, sequence it all, and then use bioinformatic approaches to try and filter out the majority maternal sequences and identify whether those fetal chromosomes are altered in their copy number or not. So generating a lot of data. But that also is the reason for the cost. So to develop a low-cost methodology, we had to work out a way of pre-analytically separating the fetal DNA from the maternal DNA, so that we weren't looking at this maternal DNA, which is the vast majority of the material, but just looking at the fetal DNA. So we've developed an approach where enzymatically we can actually chop up the maternal DNA, leaving us with an almost pure fetal DNA template that we can then amplify and then compare those chromosomes 13, 18, 21 copy number to the other chromosomes to determine if those chromosomes are imbalanced and therefore no longer 2 copies, but 3 copies.
So basically the method rely on different state of methylation. Is it correct?
It is a differentially methylated approach, yes.
Okay.
Yeah, so where the fetal material is methylated, the maternal material is not, and therefore we can inactivate the maternal material prior to the reaction based on that.
Like basically, how good is this method? I mean, as far as I know, the percent of fetus DNA varied quite significantly in the plasma. So, do you have any problems when it's low? Or?
Yeah, we do have a threshold of fetal DNA that's required in order for the reporting software to report a result, because as you get to very low copies of fetal DNA, you get more noise. So we've determined that threshold, and that really there's a very low number of patients that actually fall below that threshold. Fetal DNA can be anywhere from 3% to, well, even 20 or 30% has been observed. But our threshold is below that 3%, so it captures the majority of patients.
And how many markers are you using?
Yeah, so we, again, using the multiplexing power, we always like to have redundancy. So we've got multiple markers per chromosome. I can't remember off the top of my head exactly how many there are per chromosome, but there's multiple differentially methylated regions on each chromosome. So we've got that redundancy in case there is some sort of rare SNP or something underneath one of those. We've got 4 or 5 at least on each chromosome.
And maybe a bit into detail, because I have to say that I'm not 100% sure that I understand how it works, how basically MALDI-TOF works in terms of copy number variation. I can understand how it works when you just detecting the SNP.
Yeah, okay. All right, so the process itself is we get rid of the maternal DNA and we're left with the fetal DNA, those differentially methylated regions that have come from the fetal DNA. And then what we do is we amplify those regions in the presence of an internal standard. So that internal standard is a synthetic template with just a single base change. And we know how many copies of that internal template are in there. So then we can work out how many copies of that region from the fetus is present in the reaction. So then we say, okay, well, we've got 600 copies of the chromosome 21, but we've only got 400 copies of all the other chromosomes. So that means that they're imbalanced and we've gained an extra copy of the chromosome 21. And it's by working out the absolute number of input molecules that went into that reaction and then saying, okay, well, my chromosome 21 has more copies than my other chromosomes. Therefore, I've got an issue, I've got an imbalance. So
basically you are measuring copy numbers of each chromosome.
Correct, of our target chromosomes, so 13, 18, 21.
But are you comparing those numbers to the other chromosomes?
To autosomal regions on other chromosomes, yes.
So are there any other limitations to the method? Probably twins pregnancy is a complicated case.
Well, we haven't validated the approach or done any work, in fact, in multiple pregnancies. In theory, multiple pregnancies, they could work, but until we actually, until we validate that, I can't make any claims based on that. These, multiple pregnancies with aneuploidies are incredibly rare. So, trying to get control materials in that instance is difficult. So that's something that we can look at as a post-market surveillance activity. But as for other limitations, it's not based on SNPs, therefore it's not specific to ethnicities. It's a differentially methylated approach in conserved regions across all populations that aren't in copy number variation, at least in the normal population, apart from, of course, the big copy number variation of trisomy 21, 13, and 18. So no, there's really no other limitations.
Are you planning to move somewhere beyond those trisomies? Basically, some maybe genetic markers, heritable diseases in the NIPT area?
Yeah, it's all possible. So, you know, I could envisage that we could add sex chromosome aneuploidies. We could potentially add paternally inherited dominant disorders. You know, that all could be added in the future. At this point, we don't have development plans in that space. We became a little bit occupied last year with that virus we talked about before, nasty little thing, and that sort of meant we had to focus very much on that. So we are focused on our chromosome 21, 13, 18 panel at this point in time, but of course there are opportunities to do further work in the future.
Am I correct that so far those tests are in the development and are not basically not using in the clinics?
No, they're in a validation phase. So Agena's done our internal validation. As I mentioned, you know, 2,000 samples have been run that have known outcomes by ourselves, and then our partners are doing their local validations, because of course in the local environment there's other pre-analytical variables that you need to validate for. You know, different extraction kits, different transport mechanisms, different collection tubes, so on and so forth. So our partners are currently in their validation studies.
How you generally feed such a new product to the market? Basically, you have to, as far as I understand it, you have to convince the insurance companies that they have to pay for the new tests. So you have to convince that it's better than whatever ultrasounds or biochemistry. How it works? How difficult is this process?
Yeah, so well, every regulatory region has its own characteristics. No country is the same. No regulator is the same. So there are things that you need to prove. So you need to prove clinical utility, sensitivity, specificity, limit of detection. You know, they're all the standard things that every regulator wants to see. And then, of course, you need to prove that it has a cost advantage to the healthcare system. And that's really where our technology benefits the healthcare system, because we're substantially cheaper than existing methods. But then we always reflex through to the existing method. And do confirmatory. But even in doing that, we're still cheaper than what was the original approach.
Can you compare the costs of NGS NIPT and Agena NIPT, like relative number, or it's a tough question?
Well, it's really hard to quantify, because what is NGS NIPT? Which particular technology, which platform? But generally, you know, our test is in the order of 5 to 10 times cheaper than what would be a standard NGS, and sometimes it's even more cheaper than that, depending on the circumstance.
Mm-hmm. And in terms of multiplexing, so as far as I understand, you can use 384 well plates, and would your NIPT test fit in one well, or so how many markers are there? So it's
a one well PCR, because you want to maximize your input into the PCR reaction, and then it's 4 extension wells. So basically on a 384 well plate, we have 96 samples split across the 4 extension wells. So 96 samples per plate. And then, of course, in a standard working day, we can do up to 8 plates. So that's near enough to 800 samples. If you want to go into a 24-hour working day, then it's substantially higher than that.
Just out of curiosity, do you have any numbers on how many instruments let's say, for example, now in Russia?
Well, I know there's a number. I know there's a number, and in fact, as the head of scientific affairs, one of the things I do is look at all the publications that are generated off the instrumentation. We're well over, I think we're well over 8,000 publications in total of the MassARRAY instrument. And recently there's been some very interesting publications coming out of Russia. So I'd like to thank our Russian users because there's a lot of publications coming out. I don't know exactly how many instruments there are in Russia, but there are a few.
And when so basically you are now talking about clinical studies. Clinical publications.
Well, these are peer-reviewed manuscripts across a variety of different applications.
We are starting with, in terms of NEEP, we are starting with blood. So yes. How hard is it to process this material up to the instrument? So how much time will it take? And
yeah, so we're looking at a 10 ml blood draw. So 10 mils of whole blood collected into a cell-free DNA stabilization tube. So the most important thing here is that when you take 10 mils of blood, there's around about 1,500 copies of cell-free DNA per mil, and there's around about 100 million copies of white blood cell DNA per mil. So big difference there. And if you don't make sure that you don't break open those white blood cells, then you're going to have a serious problem. So we take 10 mils of blood into a cell-free DNA stabilization tube. At that point, that can be put in the mail and sent to the laboratory. And it needs to be processed within 3 to 7 days from that point, isolating the plasma, extracting the plasma, extracted DNA goes into the reaction, and then it's processed over the course of that 1 working day.
If I understand you correctly, so you have about a week before you, between sample collection and the time point you have to done the assay. So
that's if you need it. Yes, that's if you need it. Ideally it won't take a week to transport to the laboratory. Ideally it'll take a much shorter amount of time than that. But I'm also aware that Russia is actually a bigger country than my country, and there's a lot of remote areas. So you do have the ability to send samples from remote areas, and you've got up to a week in order to send those samples.
And I'm also not familiar with this. Do you need to keep those samples like whatever on ice or
just room temperature?
And what stages of pregnancy you can do your tests, like how early you can do it?
Yeah, so our cohorts have all been in the in the 9 to sort of 16 week area. That's the area where we're focused on. We would have to validate for timelines outside of that. Of course, later in pregnancy, I don't think will be an issue, but it's less informative later in pregnancy. Earlier in pregnancy would need to be validated.
So are you planning to do it? Because probably as early as, the earlier the better, at least as I understand it.
Well, yeah, and that's running a clinical study, which our partners can determine whether that's needed. You know, in some areas, people don't engage with the health system, or at least not engage with the referral health system, so being obstetrics and gynecology, until at least the 9-week stage.
Could you maybe specify, just again out of curiosity, in which countries those tests are going to be launched?
We're working with a variety of different countries. I won't name them all at the moment because they're in various stages, but obviously we're working very closely with Russia.
Basically, I'm probably out of questions on NEEP. If you have any ideas what I might have forgotten, some important points.
Yeah, well, I think we've covered a lot today. Yeah, I had some thoughts that I wrote down prior to meeting with you, and I mean, really, the only other area we can talk about is the analog of NIPT, and that's looking at cell-free DNA in cancer. And we have a variety of different applications there. It becomes incredibly more difficult in cancer because the fetal component, as I said before, is about 3% or more. You're not in cancer, it's much lower than that. So we're doing a lot of work. We're taking all our learnings from NIPT and applying that over to cancer. And we've got some exciting work happening in that space as well.
Actually, I wonder, like, okay, in NIPT, I do understand how you can measure the copy number variation because basically it's a stable copy number variation. In cancer, I suppose you measure basically copy number per SNP. And you have different minor allele frequencies, and it's become like increasingly difficult. How you cope with this?
Yeah. So copy number in cell-free tumor DNA is incredibly challenging. But a lot of the work that we're doing in cancer is more gain of function mutations, specific point changes, small insertions and deletions that are activating oncogenes and causing uncontrolled cell proliferation. So the mechanism of cancer is very different. So it's a different type of biomarker that we're looking for there.
Actually, I quite interesting. For example, you are testing for a specific mutation. How sure you are that you actually caught this mutation? So okay, basically it becomes quite important when it's connected to the patient. You have to guarantee the guy that you basically caught it, or it either have it or have not. And I know that it's quite difficult in sequencing, for example, because you need to be well above the coverage threshold.
Yeah. So again, yes, there's challenges in sequencing because of artifacts and other things, and there's complex bioinformatic approaches and unique molecular barcodes or identifiers that are utilized. With our technology, it's different. We don't have those sort of sequencing errors that you observe elsewhere. So we have high levels of sensitivity and particularly specificity provided by that three-level approach of PCR, single base extension, and then measuring the molecular weight with the mass spectrometer. And a mass spectrometer takes 400 measurements in seconds. Your eyes can't actually see how fast that laser is flashing and creating measurements. It's 3 nanoseconds in order for it to fly through the flight tube. Those three levels of approaches give us a high level of confidence. Now, most of the work that's happening at the moment is, really either at time of diagnosis or during treatment monitoring, and often it's in advanced disease. And I think there's a lot of opportunity to really move it into earlier stage disease, which is much more challenging. But we've got some interesting data that we're working on at the moment and will likely publish later in the year in that space.
Yeah, so basically my question was a bit like how rare cells you might catch, because tumors are extremely heterogeneous. So you might be interested in super low MAF mutations, which might become important later after the treatment, which got amplified. Like, can you put any numbers on this?
Yeah, so our sensitivity with our ultraseq chemistry, our sensitivity is 0.1% or 1 in 1,000. Which I think I mentioned before, the number of cell-free DNA copies per mil is around about 1,500. So it's really, getting down to the limit of the amount of input that you're putting into the reaction. But you're right, it needs to be amazingly sensitive from an analytical point of view to detect those, single molecule mutations.
The size of your oncopanels, like how many mutations you basically can detect in the circulating free DNA?
Yeah, so let's take our ultraseq lung panel, for example. That's the 5 main genes in the guidelines. So EGFR, BRAF, KRAS, PIK3CA, and ERBB2. So the 5 genes, around about 70 mutations across those 5 genes.
Okay. And as far as I understand, you do not clean your tumor DNA from the other DNA. So on contrary to what you are doing in NIP, so you just collect all the free DNA you have and run your tests on it.
Well, that's right, but the methylation approaches that we utilize in NIPT is an opportunity for development in cancer, and we've got some work in that space. Okay, so you also might use methylation state of cancer later in your research. Correct. I see. That's actually interesting. Okay, so now I think that I'm definitely out of questions. Of course, we can probably talk more about cancer, but it's probably let's save that for another discussion.
That's a different topic. Okay, then I probably want to thank you very much for this interesting talk, and I definitely will be looking into the area of NIP and Agena later. So we'll monitor this area. Great, thank you, Eugene. It's been a pleasure to talk to you.